Welcome back, dear students. So, start with the today's topic, which is a DNA replication. What is DNA replication? It's converting a single DNA helix into double identical copies, or parental DNA into daughter cells. Described as a semi-conservative replication. As you can see in this given diagram, this is double-stranded DNA, or you can say parental DNA. When it binds, it's split into two strands, and the template this DNA uses as a template DNA, and newly daughter DNA synthesized using this template DNA. And this process is known as DNA replication. DNA replication involves an incredibly sophisticated, highly coordinated series of molecular events, and these events are divided into four major stages. Like, first one is initiation or unwinding of DNA, primer synthesis, and elongation. So start with the initiation. Several enzymes and accessory proteins involved in DNA replication. So start with the initiation and unwinding. During initiation, or initiator protein bind to replication origin, or replication always start with the specific point, which is known as origin of replication, or ODC. So, specific base pair sequence of nucleotides are mainly present in origin of replication. This binding triggered the process unwind the double helix DNA. As you can see, there is an opening of this double helix DNA, and into the single-stranded DNA. Several groups of protein are involved in this unwinding. For example, DNA helicase, which is a major enzyme, and DNA helicase are mainly responsible for breaking the hydrogen bond that join the complementary nucleotide bases to each other, and these hydrogen bonds are essential for double-stranded DNA formation, because newly unwound single-stranded have tendency to rejoin another group of protein, or SSP protein, mainly bind with this single-stranded DNA and keep the single strand DNA stable until elongation begins. A third family of protein, which is known as topoisomerase. So what function of topoisomerase? Topoisomerase enzymes mainly reduce the some torsion strains. As you can see, highly compressed double-stranded, they have some torsion strains, and the caused by unwinding of double helix. This topoisomerase unwind this torsion strand or relieve the pressure between these bindings. As DNA strand begin to unwind into separate single-stranded is known as origin of replication. And when the double helix unwind, replication process proceed along two single strand, as you can see, in opposite directions and single but opposite direction. This form of two replication for that move along with DNA replication as they go. And after that start the primer synthesis. Why? Because primer synthesis mark the beginning of actual synthesis of new DNA molecule. Primer are short, as you can see in this green color. Primers are very short stretch of nucleotide, around 10 to 12 bases in length, synthesized by RNA polymerase enzyme, the enzyme responsible for actual addition of nucleotides to the DNA strand, and it can only add TIC ribonucleotide at 3' end of 3' OH group. As you can see in previous, this 3' OH group. And what happened? Existing chain cannot be synthesized de novo. And primase enzyme on the other hand can add ribonucleotide. Later, after elongation is complete, the primer is removed or replaced with DNA nucleotides. And after initiation, elongation start. Finally, elongation, the addition of nucleotide to the new DNA strand. What happened during the elongation? Synthesis of the growing strand involved adding nucleotides one by one in the exact order specified by the original template strand. And that Watson, according to Watson and Crick DNA model, is that adenine is always bind with thymine and or always paired with thymine with double hydrogen bond, while the cytosine is always paired with guanine. As you can see in this diagram, cytosine always paired with guanine. So for example, if the original strand read AGCT, the new strand will be TCGA, and DNA is always synthesized in 5' to 3' direction. That nucleotide always added only to the 3' end of growing strand, as you can see in this diagram. So DNA is always a you can see, as scientists have identified that polymerase then can add bases to the 5' end of the DNA strand. And if we talk about DNA polymerase, so while studying E. coli bacteria, author Korenberg discovered the DNA polymerase, which polymerase catalyzed DNA synthesis, and Korenberg discovered the enzyme DNA polymerase. And there are mainly three different DNA polymerase. For example, DNA polymerase third does most of the elongation work or polymerization, adding nucleotides on the 3' end and newly adding the nucleotide at 3' end. Other enzyme including DNA polymerase 1, DNA polymerase 1 and RNase H are responsible for removing the RNA primer. And after removing RNA primer, DNA polymerase has begun its work and replacing it with the DNA nucleotides. When these enzymes finish, they leave the nick between the section of DNA and the primer and elongation section of DNA. Another enzyme which is known as DNA ligase. Ligase enzyme mainly fill the gap. So DNA polymerase only moves in one direction. After the primer is synthesized on the strand of DNA, DNA strand unwind and synthesis and elongation can proceed in only one direction. As previously mentioned, DNA polymerase can only add to the 3' end, so 5' end of the primer remain unaltered, and consequently synthesis proceed immediately along with the leading strand. As you can see in this red color, this is leading strand, and replication along it is called as discontinuous replication. There is no breakage between this strand. This is continuous strand. This is known as continuous replication. And in another side, you can see in this pink band segment, which is known as Okazaki fragment. This discontinuation replication takes place in lagging strand, and these fragments are then filled by the enzyme known as ligase enzyme, and the fragments of newly synthesized DNA along the lagging strand are called Okazaki, and the name of honored discoverer of biologist Reiji Okazaki. So Okazaki and his colleagues made their discovery by conducting what is known as pulse chase experiment. So the DNA strand continue from 5' to 3' direction. Enzyme DNA ligase used to fill these Okazaki fragment gap with nucleotides. So when DNA replication is completed, two molecules are mainly formed because half of each strand is new and half of original is called semi-conservative replication. So this blue line is old DNA strand and synthesized two half of this is original DNA and this is newly synthesized DNA from old DNA strand, and this pink one is also newly synthesized DNA from this old one, and this is known as semi-conservative replication. Thank you so much, dear students.